Welcome guys. Today we will discuss another cell organelle of the endomembrane system, that is the Golgi apparatus or the Golgi body. This cell organelle was discovered by an Italian cytologist known as the Comoli Golgi. Comoli Golgi discovered this cell organelle, and this cell organelle was named in his honor as the Golgi complex or Golgi body, or sometimes as the Golgi apparatus. He received a Nobel Prize for his work on discovery of the Golgi apparatus. Later on, two more scientists, that's the Dalton and Flax, they came and they researched upon the ultrastructure of this cell organelle, and they gave the detailed structure of this cell organelle. Sometimes this cell organelle is also known as the Dalton complex. Now we will focus on the structure of this cell organelle. As per the Dalton and Flax, this cell organelle is basically, it has three components. This is the first component of this cell organelle. These four bow-shaped structures you see over here, these are the cisternae of this Golgi complex, and these are the vesicles. These structures are the vesicles, and similarly there are certain other structures which we call as the tubules. Now these cisternae have their origin from the endoplasmic reticulum. At this phase, that's at the lower phase of this cell organelle, you will see certain small vesicles over here, these vesicles. And these vesicles, basically these small vesicles you see over here, they pinch off from the membrane of the ER, and later on they fuse and they form the cisternae of the Golgi apparatus. So we shall understand over here that the Golgi apparatus has its origin from the endoplasmic reticulum, as 10 small vesicles pinch off from the endoplasmic reticulum, which later on join to form the cisternae of the Golgi complex. These vesicles and tubules emerge out from these cisternae of the Golgi apparatus. After that, we have to see that these cisternae are also interconnected with one another. Now, after discussing the structure, we shall move towards the concept, that is the polarity of the Golgi apparatus. We say that this cell organelle has a very important characteristic feature that's known as the polarity. What do we mean by the polarity? We see that this phase of the Golgi apparatus is known as cis phase, and this phase is over here, it is known as the Tronus phase, and this is the medial phase. So this convex phase on the lower surface, this convex phase is known as the, it is known as the cis phase, or sometimes it is known as the formation phase or formative phase, because the new cisternae are added from this side. That's why it is known as the cis phase or formative phase. Whereas the mature cisternae, they move upwards. That's why this is known as the Tronus phase, or it is also known as the maturation phase. The phase between the Tronus phase and the cis phase, or the region between the Tronus region and the cis region, is known as medial phase, which you also can call as the medial region. After that, you see that the number of cisternae, it varies. The number of cisternae usually is 6 to 12. Sometimes it can be different, but in case of the fungal cells, you see that there is only a single cisternae. So in case of the fungi, there is only a single cisternae in the Golgi complex. That's why we call the fungi as having the unicisternal Golgi apparatus. That's, it has a single this cisternae in it. So this is the structure of the Golgi apparatus. Now after that, we shall move towards the this functioning of this very cell organelle. The first important functioning of this cell organelle is that it is known as the processing plant of the cell. The endoplasmic reticulum, particularly the rough endoplasmic reticulum, it synthesizes in it the proteins, and the proteins are packed in the vesicles. Then they move and they join the cis region of the Golgi apparatus. From this region, you see these proteins are processed, and after the processing of the proteins, for example, the proteins are processed in a way that is, sometimes we need glycoproteins, and for that purpose the carbohydrate component is added to the proteins, and all this happens in the Golgi apparatus. And this glycosylation process, that's the addition of the carbohydrate to the proteins, it occurs with the help of enzymes which we call as glycosyl transferases. Sometimes also see lipolysation also occurs over here, means that certain proteins are they are what we call as converted into these lipoproteins, that some lipid component gets lipid component and the protein component, they form the lipoproteins. Similarly, after the this processing of the proteins, that is after the conversion of the proteins into the different categories of the proteins, they are differently sorted in the cisternae, which is known as the protein sorting. So first is protein processing, after that there is protein sorting, and after that with each protein there's attached a signal peptide, and this signal peptide decides the destination of the protein as to where it has to go. Because once the proteins are secreted from this trans region of the Golgi apparatus, they go outside the Golgi, and many of the proteins are needed in different cell organelles inside the cell, but some proteins are also needed outside the cell. This signal peptide which is attached with the proteins, it determines where the destination of the secretory protein lies. So this process of adding the signal peptide to these proteins is known as the tagging process. After the tagging, there occurs the secretion, that is these proteins are secreted. So the first and foremost function of the Golgi apparatus is the protein processing, protein sorting, tagging, and secretion. That's why Golgi apparatus is known as the processing plant of the cell. After that, sometimes we also call these Golgi apparatus as the post office of the cell, because in the post office there are different letters and they have different addresses where they are to be delivered. In the same way, the proteins processed over here are to be attached with the different tags, so that they can be sent to different destinations inside and outside the cell. This Golgi apparatus has a very important role in the formation of the cell membrane. Also, the carbohydrate component of the cell membrane is basically it is derived from the this Golgi apparatus. After that, there are some functions of the Golgi apparatus, like the formation of the lysosome. Some vesicles pinch off from these structures known as Golgi apparatus, and those vesicles contain the hydrolytic enzymes, and those structures are known as the lysosomes. So the lysosomes also emerge out from the Golgi apparatus. One more function of the Golgi apparatus is its role in the formation of the plant cell wall. The components of the plant cell wall, that is the hemicellulose and pectins, they are derived from this Golgi apparatus. So the Golgi apparatus is a very important component of the endomembrane system, and as per the Novikov, the endoplasmic reticulum, Golgi, and the lysosomes togetherly form a system which we call as Garel system, GERL, Garel system. G stands for Golgi, ER for endoplasmic reticulum, and L for lysosomes. So this is the main functioning of the this is the main functioning of the Golgi apparatus. I hope that you have understood the detailed structure and the functioning of this important cell.